Hello and welcome to DNA Today. I'm your host, Kira Deneen. DNA Today informs you on what is happening in the genetic world. The show's packed with news stories, lessons, all to keep you updated and educated on the ever-changing world of genetics. And today, it's full of news stories. I've got a lot to cover in only half an hour to do it. And today I'm talking about 2 out of 3 of the stories are about genes and discovering new genes associated to different disorders and diseases and cancer. A new study finds gene variants may be contributor to PTSD. New genes linked to triple-negative breast cancer is another one, and scientists have successfully used light to trigger REM sleep in mice. And so we'll get into what REM sleep is and how scientists were able to use light to trigger a certain stage of sleep, which is really interesting. And it's cool to see how they go about actually doing this in the lab. So before I jump right into the news, I want to mention that my time live on the radio will be moving to 11:30 a.m. instead of 1 p.m. It's still on Mondays. You just gotta listen a little bit earlier, 11:30 a.m. And this will take effect January 26th, so not next week, but the following, once the semester kicks off. So again, this is 11:30 a.m. instead of 1 p.m., still Mondays, and this is going to take effect January 26th. Of course, I will remind you this next week, but just so you can put it on your calendars if that's something you do. All right, first news story. So I mentioned that genes have been are found to be contributing to PTSD and the risk that PTSD has in different people. So new research from UCLA explains that PTSD risk could be increased by certain genes, or gene variants. Now, PTSD stands for post-traumatic stress disorder, and some people have this disorder as a result from a traumatic experience. Now, this genetic research gives an explanation on why some people can develop PTSD while others don't, because not every soldier that comes back from war does have PTSD, even if they're in similar situations and had similar experiences. So this research really looks at what the difference is, why some people get it and others don't. Now, it's not the full explanation of exactly why it's happening, but it's some more information on understanding why there is a difference. Lead author of the study, Dr. Armin Gioan, explains this concept, quote, "Many people suffer with post-traumatic stress disorder after surviving a life-threatening ordeal like war, rape, or a natural disaster, but not everyone who experiences trauma suffers from PTSD." End quote. And UCLA's genetic research team identified 2 gene variants linked to an increase in the likelihood of PTSD affecting an individual. So with these new gene variants identified, it could lead to a better diagnosis and treatment of PTSD patients in the future. If we're able to do a gene test and say, yeah, actually you're predisposed to getting PTSD if exposed to a bad experience, if you have a bad experience or traumatic experience rather, that's very important that it's a traumatic experience, that, okay, you have a higher risk that you are going to develop PTSD where this other person doesn't have the gene variant, so they don't have as high of a risk. And again, it's good to stress that this is not the full explanation, that there are obviously other things that go into this, but it's we're exploring this more in depth of what is really contributing to PTSD genetic-wise, and it'll be interesting to see in the future what percentage of it is genetic and what percentage of it is maybe environment, how you've grown up. That nature versus nurture always debate, and it's a debate, but it also goes hand in hand, that it is a combination of those 2 forces coming together. So let's look a little bit more into what PTSD is. The National Institute of Mental Health reports that 6.8% of the adult population of the U.S. experience PTSD at some point in their lifetime. 6.8%. So 6.8 people out of 100. Now, the condition can lead to several debilitating symptoms such as anxiety, avoidance of people, places, or situations, memories coming up, loss in concentration, negative changes in thinking and mood, and many symptoms can be triggered by the onset of something that reminds the patient of the traumatic event they experienced that brought on the PTSD. So it could even be a certain color that maybe they were exposed to this color while something was happening, and now they see that specific color and they start feeling these symptoms again. I just thought of the, now I can't remember what movie it was, but it was a movie about multiple personality disorder, and and now I'm forgetting the actor, but I'll remember for next episode and say that. But she kind of had a lot of PTSD, and she kind of had multiple personality disorder from that. And a famous case of research on PTSD was back in 1988 when the Armenian earthquake occurred. And this was a huge earthquake, very, very tragic and sad. Scientists, also from UCLA, studied the DNA of survivors of the tragic, tremendous earthquake, and these samples have been studied since 1988. Research in 2012 involving these samples led to the discovery that survivors carrying 2 gene variants associated with depression were more likely to have PTSD than those that did not carry the variants. So, research just 2012, 3 years ago, found this relationship between people that these 2 gene variants are associated with depression, but they also are more likely, the individuals that have those gene variants are more likely to develop PTSD than those that did not, because they were able to have a large sample of people that went through this traumatic experience of the Armenian earthquake and could see what actually happened to them and what their genes have, and if there was a relationship between that. So these gene variants I keep referring to, let's get into like what they are and what they do and what they're called and all that. So the first one, they're called COMT and TPH2. Now the first one, COMT, is a gene for the enzyme that degrades the mood regulator dopamine. So it's this gene produces a protein, because an enzyme is a protein. It produces a protein that's degrading a mood regulator molecule called dopamine. And you've probably heard of dopamine. Last episode, I had a whole news story about dopamine. The second gene, TPH2, is a controller of the production of serotonin. And this is another mood regulator molecule. And so this TPH2 is regulating how much of the serotonin is being produced. So these are 2 mood regulators. They're both dopamine and serotonin are related and linked to mood and happiness levels. A lot of times in depression, these levels will be lower than normal, and that's, people think, depression, it's just kind of your environment, whatever, but a lot of times it actually is your brain chemistry, and you may not have enough dopamine and serotonin in your brain to make you feel happy, a normal content level of happiness. So these 2 molecules are very important in regulating your moods. And so if these are affected, then obviously your moods are affected, and that's where depression and PTSD can come into play. The lead author of the study I mentioned before, Dr. Armen Jogian, comments on the gene variants. Quote, "We found a significant association between variants of COMT and TPH2 with PTSD symptoms, suggesting these genes contribute to the onset and persistence of the disorder. Our results indicate that people who carry the genetic variants may be at a higher risk of developing PTSD." So kind of what I've said before, but he puts it into more elegant terms. And so he's really saying that these 2 gene variants are linked to a higher risk for PTSD. So again, that is huge, because we could test for these gene variants and make a huge impact on, okay, you have a higher risk, and so, we could prevent you possibly, like for soldiers, for instance, there's a lot of testing that they go through to be in the Army, to be in the Navy, the military, and so this could maybe in the future be a test that if you have a high risk for PTSD, that you have these 2 gene variants, maybe they won't take you, or maybe they won't put you on the front lines or something of the sort. So this really could come into play not just in the medical field, but in society, in different places, that people that in their jobs are exposed to traumatic events that maybe they wouldn't take those jobs if they know that they have these gene variants and are at a higher risk for PTSD. And so another question to pose is knowing how the gene variants affect medical treatment of PTSD. So could this knowledge even lead to predicting if someone's vulnerable to developing PTSD? And that's kind of the hope. Dr. Armen expresses that his hopes for this new information could be used to develop screening procedures that would identify people that are at risk for developing PTSD. And he also mentioned the treatment side of PTSD, saying he wishes that the technology could be developed to identify new forms of drug therapy to prevent and treat the disorder. So I've talked a lot about preventing and screening beforehand, but if someone has PTSD, maybe knowing about these gene variants, we could develop drug therapy, who knows, maybe even gene therapy, to treat the disorder once someone already has it. Because if we find that these gene variants kind of allow for PTSD to develop, then maybe if we change these gene variants and kind of alter them to be more of the normal a normal version of those genes, that maybe the PTSD symptoms would ease up a little bit. Who knows? This is kind of speculation on my part, but it's very interesting to see what could come from all of this information, and it's kind of a springboard, as a lot of these news stories are, of where we can go from here, medically, research-wise, really expanding our knowledge to be able to really help people and treat different disorders. So he expands on these ideas, saying, quote, "A diagnostic tool based on PTSD-linked genes would greatly help us in identifying people who are at high risk for developing the disorder. Our findings may also help scientists uncover more refined treatments, such as gene therapy or new drugs that regulate the chemicals associated with PTSD symptoms." So again, kind of what I just said, but really it's focusing on if we identify these genes that maybe we really can target them and change them to then change the PTSD symptoms. And imagine if we were able to predict, who would get PTSD and how that could prevent, in many cases, the number of people affected by it. Prevention is obviously key, because if you can prevent it, you don't have to treat it and everything. You're just eliminating all of that. And although 2 gene variants have been discovered that are linked to PTSD, Dr. Armin Geoghegan is reserved in predicting that there are most likely more genes that PTSD is caused by. So, if this study can expand and find more of these gene variants, and maybe gene therapy can be used for victims of PTSD, that could be great, because if you only have 2 gene variants that you're targeting for gene therapy, it's not going to be as good as if you, say, have 4 or 5. So I'll definitely keep you updated if I see any more news about PTSD and the genes that kind of associated with that. Next news story is talking about breast cancer, and I've talked about breast cancer many times on the show before, discussing Angelina Jolie's story of genetic testing and the double mastectomy she had, and also just being very, very out in the public and kind of educating the public how important gene testing is and how important genetic counselors can be in helping patients in this time of figuring out the genetic testing, because they are the experts in genetic testing. And she has been kind of in the public eye for this and really trying to push people to go for genetic testing, and not necessarily to, go for surgeries. Like, that was her personal choice, but really to just be more educated so you can make educated decisions on what is happening to your body and, taking control over that. I also talked about October being Breast Cancer Awareness Month, and I've said more knowledge on the BRCA gene that are heavily linked to breast cancer. And I believe I will be talking about BRCA gene, maybe not, we'll see. So now scientists have identified new genes that is linked to driving triple-negative breast cancer. Now, I didn't know what this was before the show, but kind of did a little research on it. Triple-negative breast cancer is when cancer growth does not involve estrogen receptors, progesterone receptors, human epidermal growth factor receptors, any of those kind of hormone receptors. So since it doesn't really involve any of these hormone receptors, therefore the triple-negative breast cancer does not respond to hormone therapy. So this means patients that have triple-negative breast cancer have more negative prognosis compared with patients with other types of breast cancer. And there are different types. The most common form of triple-negative breast cancer is basal cell breast cancer. Now, scientists are investigating new ways to treat triple-negative breast cancer, and much of this research has been focused on identifying genes that are linked to triple-negative breast cancer, as a lot of these stories are. If you can identify the genes that are really related to the disease or the cancer, then you can focus on using gene therapy for those and kind of expanding your knowledge from there. So it's a good place to start to look at what's really happening genetically and how we can kind of manipulate that for treatment and maybe even for a cure someday. So it's great that they have identified some of these genes, but it hasn't been super successful as it's only a few genes that have been identified. It's not really a whole slew of genes. And a new study by Dr. Khalid and colleagues took a different approach than normal to identify these genes. So it's kind of cool. They assessed alterations to genes that influence the action of stem cells and developing tissue. And as past research from the team indicated that these alterations affect cancer development. So they're focusing on genes that are influencing the actions of stem cells and developing tissues. So they're really looking at cells that proliferate very well, that they're growing, expanding, multiplying really well. And that's what stem cells and developing tissues are. They're kind of, you can think of them as kind of like baby cells that, your adult cells, they're kind of doing their thing, but, as babies, you're growing rapidly. So again, they're looking at stem cells and developing tissue. Now, the team found that a gene called BCL11A was particularly active in triple-negative breast cancer. And this was after studying gene alterations in around 3,000 patients. So they definitely had a lot of patients to work with to really find and figure out what gene was associated with triple-negative breast cancer. So wanting to study the BCL11A gene's impact on breast cancer further, researchers studied how the gene, when turned on, reacted to human and mouse breast cancer cells, or breast cells. We'll see if they're cancer. So they're taking this gene that they think is associated with triple-negative breast cancer, and then they're turning it on and seeing how the cells react, seeing if they are cancerous, seeing if they're not cancerous, and kind of seeing how much this gene is impacting the triple-negative breast cancer. So the results did support the idea that the BCL11A genes have an impact on breast cancer, as the cells being tested with the BCLA11 genes turned on started acting like cancer cells. Just as I said, when you turn this gene on, then these breast cells started acting like breast cancer cells. And they used human cells as well as mouse cells. Mice, you'll hear me in almost every research story talk about mice because they are really the best organisms to work with and test things because they're so close to humans. I think it's like, I want to say 88% of their genes are the same as humans. Don't quote me on that, but it's something around there. And they are small, and so they're easy to, keep in labs. They have a pretty fast reproduction time, and there's a whole nother list of things that make them great organisms to work with. But don't be surprised every time you hear mice. You probably think of that like a lab. You have mice in there, whatever. But yeah, mice are definitely the best organisms for a lot of studies. Not every study, but they're a pretty universal organism to work with. So I said that once you turn these genes on, these breast cells turn into breast cancer cells. So really, you could see the correlation of this gene, when activated, is really contributing to these breast cells turning into breast cancer cells. Now, the opposite occurred too. And don't think the opposite, what does that mean? So when BCL11A activity was reduced in human triple-negative breast cancer cells, the cells lost some of their cancer-like characteristics. So you can think of, a gene like on-off. So when the gene switch is turned on, it's not actually switch, but just imagine, when the gene switch is turned on, cancer is happening in these breast cells. Now, when the switch is turned off, these cancer-like characteristics are kind of disappearing. So obviously this gene has a tremendous impact on the cell being cancerous or not. And in mice, these cells were less likely to drive tumor growth. And tumor growth is obviously a cancer-like characteristic. So when the gene was turned off, it was less likely to drive that tumor growth in mice. Now, Dr. Khalid sums this up nicely. Quote, so by increasing BCL11A activity, we increase cancer-like behavior. By reducing it, we reduce cancer-like behavior. So very straightforward in explaining this concept of when the gene is on, cancer is on. When the gene is off, cancer's off. Obviously, that's a very blanket statement, but that's kind of the idea behind this. And even more experiments were conducted that support the idea that BCLA11 contributes to breast cancer, and these experiments were again with mice. So when the rodent's BCL11 11A genes were inactivated, tumor growth was stalled in their mammary gland, and mice that did not have this gene inactivated, however, developed tumors. And a study author of this research, Professor Carlos Caldas of University of Cambridge and the Cancer Research UK Cambridge Institute, said, quote, "This exciting research identifies a novel breast cancer gene in some of the more difficult to treat cases. It builds upon our work to develop a comprehensive molecular understanding of breast cancer that will inform clinical decisions and treatment choices. Finding a novel gene that is active in cancer should also help in the search for new treatments." As always, of what I say is, when we're identifying these genes, it's more exciting to see that more treatment is up ahead. All right. Next news story is talking about REM sleep and how scientists can trigger it with just some light. So this kind of blew my mind, and I had to, once I saw the heading of this article, I was like, man, this is interesting. I definitely have to read into this. And I love learning about sleep and all of that, and the different stages of sleep and dreams and all that. So this was a cool little news story I definitely wanted to include. And so what is REM? I keep saying that. I'm not talking about the band. I'm talking about REM standing for rapid eye movement, and it's a very important deep sleep stage. Criteria for REM sleep includes rapid eye movement, low muscle tone, and rapid low voltage EEG. Now, vivid dreams are often recalled from the REM sleep because it's that deep sleep, but your eyes are moving really fast. And sometimes when you watch someone sleep, you can see that their eyes are moving really fast. Like their eyes are still closed, but you can see them under there moving. And it's really cool. And during REM, the activity of the brain's neurons is quite similar to that of waking hours. And for this reason, the REM sleep stage may be called the paradoxal sleep. Now, although we understand the significance of rapid eye movement, of being, in that restful sleep, that's the real quality sleep, we don't really understand what controls it. We know it happens, but why is it happening? What's controlling this sleep stage? And get this, researchers are using a new technology called optogenetics, have discovered they can trigger REM episodes in mice by shining a light directly into select brain cells or neurons. So this light is activating REM sleep. And the study was from a team made of members from Massachusetts Institute of Technology, MIT, and Harvard Medical School, both located in Massachusetts in the United States, and that's right around the corner from us here in Storrs, Connecticut. And researchers explained that we already know the brainstem contained Colorgenic cells or neurons that are involved in controlling REM. But because the brain region also contains a lot of other cell types, it's not easy to tease out the unique role of Colorgenic neurons. So let's back up a moment to explore what these Colorgenic neurons are. So they describe any neuron which mainly uses the neurotransmitter acetylcholine, ACH, and it's using this to send its messages. And so these neurons provide the primary source of acetylcholine to the cerebral cortex and are known for their role in promoting cortical activation during both wakefulness and during rapid eye movement sleep. Lead author Dr. Christina Van Dort of the Department of Brain and Cognitive Science at MIT explains that previous studies have suggested brainstem Colorgenic neurons are active when we are awake and also during REM sleep. But nobody could actually say whether the firing of these specific cells was responsible for the transition to REM sleep. So there was a correlation there, but we couldn't say that was the reason why REM sleep started, or REM sleep, I'll also refer to it as that. The study focused on finding out if these Colorgenic neurons could actually induce REM and not just be linked to REM sleep. And these scientists were able to use radical new technology called optogenetics that is helping scientists understand the brain's wiring by controlling brain cell activity with light. So in optogenetics, neurons are made to react with light with an insertion of a protein found in algae. So in nature, proteins respond to certain wavelengths of light, allowing algae to move around. So if you insert the protein into certain types of the brain cell, then you can shine a light on them and activate them, essentially controlling brain activity at the level of individual cells, which is really cool. You're injecting this protein, which then moves to light, and when that protein is moving, then these cells are activated. So using mice where the light sensitivity protein had been inserted in their Colorgenic neurons, the neurons could be activated via a fiber optic device mounted on the heads of the mice. And they found if they activated the light sensitized colorgenic neurons during non-REM sleep, it increased the number, but not the duration, of REM sleep episodes the mice had. Further analysis showed that induced REM sleep matched the natural REM episodes very closely. So why is this study significant? Why do we care that, if we do this special weird technique, we can induce REM sleep? Well, it's important because it provides more information about how REM sleep is controlled, and this is a step towards understanding how to design natural sleep in humans. And so far, drugs have not been able to replicate the benefits of natural sleep, where REM and non-REM states alternate every 90 minutes or so, as Professor Brown explains. Quote, what they do is create sedation. If you are lucky, the sedation allows your natural sleep mechanisms to take over. So hopefully by learning more about what controls REM sleep, we can kind of figure out how we can give that to people that have insomnia or other sleep disorders and be able to help them. That's the news stories for today. Now, I want to introduce a new segment. I'll be ending the show here with here and there, not every week, when I'm feeling up to it, called Recommendations. And this is where I recommend something for you to go check out. And this week I want to recommend a podcast. Wow, shocker, a podcast. Not mine, but another genetics podcast. And over winter break, I had a little more free time than usual and was able to listen to some new podcasts. And it was a 5-week break, so I got to a lot of podcasts. And there's so many I'll be recommending in the future. I have a whole list. But for this week, I want to recommend one that actually caught up with over break. I've been listening to it since summer 2014. It's called Naked Genetics. And this is a monthly podcast that features interviews with key researchers in genetics throughout the world. It's relatively easy to follow along. Sometimes the news stories get a little too much in detail. Not too much, but kind of more complicated than a traditional listener would be able to follow along. But sometimes you just kind of stick with it, then they move along very quickly. So if you're not understanding a news story or an interview of research that a doctor is talking about, then, just keep it together for like 2-3 minutes and it will move along very quickly. And it's really cool. They have like a gene of the week on there and different interviews. And usually the episode is focused kind of on one topic, which is cool because you get to kind of learn a lot in depth about one topic. You can check out their website at nakedscientist.com/genetics or search for the podcast in iTunes with keywords Naked Genetics. Also, if you're interested, DNA Today is on iTunes. You can search for DNA Podcast. It could also be DNA Today. I'm not sure, but you'll see my logo is an orange DNA molecule, and I believe it even says my name on there, Kira Deneen. So yeah, you could definitely check those out on iTunes. And obviously it's free because podcasts on iTunes are all free, and definitely be sure to look at all kinds of podcasts on iTunes. It is the king directory of podcasts. So again, I want to recommend Naked Genetics. You can find them on iTunes or their website. They have a lot of great stuff on there, nakedscientist.com/genetics. And they have other shows than just genetics, but I suggest the genetics one. That wraps up today's episode of DNA Today. Tune in next week, Monday at 1 p.m. on WHUS.org or 91.7 FM to learn more about genetics. After next Monday, you can listen live at 11:30 a.m. instead of 1. Again, that takes place the 26th once the semester kicks off. Martin Luther King Day, I'm still on at 1 p.m. So 11:30 is the following week, 11:30 a.m. I'm not doing the late night shift here. DNApodcast.com is full of content, including episodes I broadcast, as well as mini lessons on genetics, loads of resources to check out, and it's definitely a great place to learn more about the show and genetics. And if you are interested in social media, you can follow on Twitter at DNA Podcast, or follow me, Twitter at Kira Deneen, KIRADINEEN. If you have a question you want answered on air, shoot me an email at info@dnapodcast.com. Again, that's info@dnapodcast.com. You can also tweet questions. That's cool too, because then other people can see them. It's more of a public way of doing it, which is great. So thanks for listening, and join me next week. Again, Monday at 1 p.m. One last time, Monday at 1 p.m. A week after, it's 11:30 a.m. for the rest of the semester. And you can listen to learn more and discover new advances in the world of genetics.